Okay, welcome to the next session, and this week we're going to be looking at the control of gene expression. So some of the learning outcomes from this session. So by the end of the session you're going to be able to describe how gene expression is controlled in prokaryotes. We'll also look at gene expression in eukaryotes. Also you'll be able to outline how eukaryotic gene transcription is regulated, and as well as describe what are epigenetic changes and how are they involved in gene expression. And finally, how is eukaryotic gene expression regulated after transcription. Kind of looking back at the kind of information pathways, so there are a number of kind of biochemical mechanisms which are kind of apparently contradictory requirements for genetic continuity and evolution in living organisms. So if we take the DNA structure, the DNA structure has a double helical nature. It's suggested how the DNA may be copied. And when we look at the RNA level, so the messenger RNA, and looking at tRNA discovery, this is really how the information in the DNA is then decoded, because obviously all the DNA that's present in the genome is used as functional proteins. Now, there's a central dogma of molecular biology, which involves the kind of transferring of information. So, looking at the DNA, which is acting as our kind of the information which is coded in that sequence as nucleotide bases. That's passed on in a sequence to the RNA through something called transcription. Now, DNA has this ability to replicate, or actually to be replicated itself, but taking the flow of that information into RNA, and actually not all RNA molecules will actually end up as proteins. Some will actually act as a kind of regulatory mechanism in both prokaryotic and eukaryotic systems. Now, if we look at the RNA, the RNA is obviously the building block for our protein, and this requires information in the RNA then to be passed on to the polypeptides, or passed to the polypeptides, which is our protein, but it's never the reverse of polypeptides to nucleic acids. Okay, so going to revisit the RNA polymerase structure. So remember, in E. coli, this is a large enzyme complex. It's made up of multiple subunits. It has a molecular weight of 390,000, which has no proofreading function. So this means that there's one error per 10 to the 4, 10 to the 5 nucleotides. Now, as RNAs are actually generally degraded after use, mistakes here are kind of less of a consequence, and so they're not going to impact the organism. When we look at the structure again, it's made up of these 5 core subunits. So we have 2 alpha subunits, which are down here. They kind of function in the assembly and binding to upstream promoter elements. So we'll discuss a little bit more about these again. We also have the beta subunit, and as well as the beta prime subunit. And also what we find also here is it's called the omega subunit. So the beta subunit is the main catalytic subunit, and then the beta prime is responsible for binding to the DNA. So this is the part of the complex that actually binds to the DNA. And then the omega subunit down here, that helps to protect the polymerase from degradation or denaturation. So as you remember, RNAs are very susceptible to degradation. And the other subunit, which is called the sigma subunit, is actually buried inside this complex that actually varies by size and helps to direct the polymerase to specific DNA binding sites. So there's some common features of E. coli promoters. When the RNA polymerase binds within essentially a region which is 70 base pairs, So when we say minus base pairs, so this is like this is before the transcription start site, to about 30 base pairs, which is plus 30 base pairs after it. So so minus 70 base pairs is before the start site, and then 30 is after. And sequence analysis actually reveals two sequences which are at position minus 10 and minus 35 of the RNA site. And they're important for the interaction with that sigma subunit, which is known as, so the first sequence, which is at minus 10 base pairs before the transcription site, is known as the Tata box, which has a sequence from 5 prime. So this is like the beginning of the sequence, and that's the end of the sequence. Remember, the 3 primes at the end of the sequence. So we have a T, A, T, A, A, T. And then the other sequence, which is at position 35 base pairs before the RNA start site, this has a sequence, it's a 5 prime sequence of TT, GACA. And certain nucleotides which are common, so these are certain nucleotides as known as consensus sequences. So these are always the same. So so these are always present at minus 10 position and also at minus 35. So we always find these sequences before the RNA start site. There's also additional AT-rich elements present, and this is normally present upstream, so at about minus 40 and minus 60. So this is before that transcription site, and this is known as the UP element, which is bound by the alpha subunit. Okay, so there are some common features of E. coli promoters. Now, these are examples of different types of promoters that we can find in E. coli, and they get different types. And there are some similarities. So remember, we're talking about those sequences that we find. So if we look at the kind of consensus sequence, so here we have the UP element, which we find upstream of the start site. Remember the 35, the minus 35 site, which consists of TTGACA. We have a spacer sequence, which doesn't really, it varies in length between the different promoters. So here it's like 17 nucleotides, so it doesn't really code for anything, but it's just like a gap sequence. And then we have the minus 10 region, which is the Tata box. But that it's not always the same in each of these promoters. So we get different types of promoters. So we've got this TRP or LAC promoter. And what happens is we get like those mutations in these kind of core consensus sequences, which actually affect gene expression. So they affect the RNA polymerase binding and initiation, and these sequences are always upstream of the RNA start site. So when we kind of look at the initiation of transcription, so what happens at the beginning is that the RNA polymerase binds to the promoter. So to that kind of promoter sequence with the omega factor bound. So here we can see the RNA polymerase core binds to the DNA promoter sequence, and this actually causes the DNA to kind of loop around. Okay, so and what happens is it's in its kind of closed complex, and in the meantime, there's like a kind of transcription bubble which forms. So it kind of creates a closed complex where the DNA and the bound DNA is intact. After this complex then opens up. So the open complex is then formed, and there's a region from about position minus 10 to about plus 2, which is then essentially unwound. So essentially this bubble starts to form. Okay. So the complex opens up, and then transcription then can be is initiated. And then what happens then is the polymerase, the RNA polymerase, then moves away from the promoter. So the promoter there's a clearance which is followed by elongation. So then the polymerase kind of moves away, and then the omega subunit then falls away and then is then replaced by another sequence known as NUS-A during elongation. So elongation continues, and then it's replaced by this NUS-A sequence. This is this like green molecule coming in to this bubble here. So you can see the RNAs. Remember that RNA primer coming out. Okay. And once transcription is complete, then the NUS-A then dissociates from the RNA polymerase, and then the RNA polymerase dissociates from the DNA. And then after, there's kind of this Zycorina recycling, so then you get the omega then can rebind to the RNA polymerase, and then binding can then begin again. So there's a regulation of transcription. So transcription is very much energy intensive. So it's actually carefully regulated process, and regulation can actually occur anywhere in the process, but it's primarily at the polymerase binding and initiation. Now, this is achieved in many ways. So there are differences in promoter sequence. So we have something called activating and repressor proteins. These actually bind to DNA sequences which are both near and also distant from the promoter. And one such protein is known as CAMP receptor protein, which is also known as CRP. This actually binds to promoters of genes which are involved in sugar metabolism other than glucose. So for termination of transcription, there are actually certain DNA sequences that result in a pause of DNA synthesis, and this may result in termination. And in E. coli, there are two types, so known as Rho independent and Rho dependent. So for Rho independent, this is actually characterized by polyUs. Okay, so we have a string of polyUs which are normally found at the 3' end of the transcript. So here we have the RNA polymerase. It's creating our bubble. Here we've got the RNA which has been made. And as RNA synthesis encounters the terminator sequence, so it comes to the end, what happens then is there's essentially an RNA hairpin which is formed at the essentially these polydromic sequence, which is reducing the length of the RNA and DNA hybrid. And so it forms this hairpin structure of around about 15 to 20 nucleotides before the end of the 3' end. And this makes the RNA polymerase then stop. So it pauses and then dissociates. And then the messenger RNA then can be released. So that's in the case of Rho independent termination. In the case of Rho dependent, so this is a little bit different. So what this involves, well, it essentially involves the Rho helicase, which binds to a root element. So this includes something called a CA rich region known as root element. And the Rho helicase associates with the RNA at a specific site, and then kind of migrates from 5' to 3' directions. Here we have the Rho helicase. It binds to a root site. Okay, so here we've got the root site, and the Rho helicase binds, and it is an ATP dependent reaction, so it requires energy, and it starts to move from the 5' to the 3'. And it does this until it reaches the transcription complex that is paused at the termination. So the Rho migrates along the messenger RNA to the elongating RNA polymerase. And there's a detailed mechanism which is not known, but it helps the polymerase then to dissociate from the DNA. So the Rho helicase separates the messenger RNA from the DNA template. So if we consider eukaryotic RNA polymerases, so we have, there are different, in eukaryotics, there are several polymerases. So we have the RNA polymerase 1, that's involved in synthesis. It synthesizes pre-ribosomal RNA, or precursors for 28S, 18S, and 5.8S ribosomal RNAs. The the RNA polymerase 2, that's responsible for the synthesis of messenger RNA. So it's just solely responsible for messenger RNA. Whilst RNA polymerase 3, this makes the tRNA, so remember the transfer RNAs that we discussed the other week, and also the small RNA products, so products like microRNAs. These are very short products of RNA which help to control gene expression. And then we also have RNA polymerase 4, which we find in plants, that appears to be involved in synthesizing or interfering RNAs. And then in mitochondria, they even have their own RNA polymerase. So there are different types of RNA polymerases in eukaryotes. So it's much more complex. So some of the features of eukaryotic promoters are kind of recognized by the RNA polymerase 2. So the eukaryotic promoters vary widely in sequence, but some have a few features in common. So remember, at the -30 position, we have the TATA box, so known as TATAA, known as the TATA box. Now, it normally has a consensus sequence of TATA, but the sequences in the 5th position could be either an A or a T, and then you have a consensus sequence of an A, and then it can either be an A or a T, or an A and a G. We also have an initiator at +1 position, so this is a sequence very close to the RNA site. So when we consider the RNA polymerase 2, it's a much more complex than the prokaryotic, than its kind of bacterial counterpart. But structurally, it's very similar. There are 12 subunits, so it's much bigger. It's around 510,000 as opposed to 360,000, wasn't it, for the prokaryotic? And it requires an array of proteins known as transcription factors. So transcription factors are very important proteins to learn about to form a kind of active transcription complex. And each step of the transcription has an associated characteristic protein. So don't worry, you don't have to learn all of these, but just to know that they exist, okay? So we have different transcription factors. We've got one which is called POL2, that has 12 different subunits. We have also the TBP, or the TATA binding protein, that specifically recognizes the TATA box. We have the transcription factor 2A, that stabilizes the binding of something called TF2B and TBP in the promoter. So we have different ones, and also we have elongation factors, known as L or Peter, etc. So they all have different roles in transcription in eukaryotes. Kind of just focusing again on transcription factors. So transcription factors are quite specific because they bind to particular regions. So you can have specific transcription factors which will bind to regulatory regions, and essentially we have general transcription factors which will bind to the RNA polymerase, which also then binds to particular sites within the DNA. And essentially they will determine whether or where the RNA polymerase II will transcribe DNA. So essentially they aid in the transcription process. And so here we've got the DNA which is bending. It allows kind of specific transcription factors to interact with the RNA polymerase complex and affect the rate of transcription. And these sequences here, they bind specific transcription factors, may be far from the actual transcription start site. So that's actually quite interesting. Kind of getting back to how transcription occurs in eukaryotes. So we get first the assembly of RNA pol II and the initiation of transcription. So we have it's initiated by the TATA binding protein known as TBP. So here we have the TBP. This binds to the TATA sequence. So TBP is part of a multi-complex such as TF2D. Where's 2FD? Here we go. Here's 2FD. And there are also other transcription factors. So TF2B. So here we are. And then we've got TF2A. And also we also have TF2F. And as well in another complex which also comes together, it's TF2E and TF2H. And so this complex is like assembled and is recruited. So pol II actually is recruited then to the DNA by the transcription factors. And this then causes then the DNA to kind of bend around, and this is known as our pre-initiation complex. And the helicase activity in TF2H helps to unwind the DNA, so create that bubble, normally at the promoter site. So after we have the binding of this RNA polymerase II and all the transcription factors, what happens is then there's phosphorylation. So this is, you might have heard about this in some of the protein lectures. Essentially, it's a type of post-translational modification. So essentially, we get phosphorylation of the polymerase at a particular region within the protein, so the C-terminal domain, known as the CTD, and this changes the conformation. So phosphorylation changes the conformation and allows then transcription. So it goes from this closed pre-initiation complex, and the phosphorylation helps to open up the complex. So once we have opening up of the complex, we get the elongation stage of transcription. So normally after 60 to 70 nucleotides, so this is at 60 to 70 nucleotides after we have something called the TF2E. So essentially here we have the TF2E. It's released, followed by then another transcription factor, which is known as the TF2H. And so we have the transcription elongation is aided by the elongation factors, and that's only after these two transcription factors are released, so they're dissociated. So the elongation factors, they bind to the RNA pol II to enhance, essentially they add to the processivity and help to then coordinate post-transcriptional processing of RNAs. And then finally, the termination of transcription. So for the termination of RNA pol II, it is then dephosphorylated. So not phosphorylated, so we remove the phosphates. And essentially by taking out the phosphates, what happens then is we get dephosphorylation. So we change the shape. So we essentially change the shape and of the complex, and then what happens then is by changing the shape, we get elongation factors dissociate, and the C-terminal domain is then dephosphorylated, and the process is then facilitated by those termination factors. So there is also post-transcriptional messenger RNA processing. So the post-translational processing, so once we've synthesized the RNA molecule, this is known as our kind of primary transcript, okay? But in fact, the job's not finished, and so we need to kind of process the RNA, the messenger RNA, and there is a further processing that occurs at this step. Now the processing includes something called splicing. So this is the splicing out of introns, and then the rejoining of exons for a continuous sequence. So here we have the 5' cap. And then we have here is the exon, and then we have this intron section, and then we have here is the exon. So during transcription we get the 5' capping, so we get this cap which is added to the 5' end, and then after we get completion of the primary transcript, this is what we see. So and so essentially it's made up of this different regions, but this is not the finished job. So what we need to do is kind of cut these sequences which are not going to be spliced out. So we need to take out the intron sequences. So the exon is the part, the information that we want is going to be read during translation. And so this is kind of cleaved, okay? So there's something called the spliceosome, which helps to splice these sequences. So there's cleavage and something called polyadenylation, and then splicing. Okay, so after kind of cleavage, there's the addition of the adding of this 5' cap, and as well as we add also some sequences at the end, so known as the poly A tail. So essentially, it's a sequence of a sequence of A. Okay, so it can have a defined length. So RNA processing, so this is the addition of a 5' cap and the poly A tail. So both the ends of the messenger RNA molecule are modified by the addition of a 5' cap, which is the modified guanine nucleotide. So here we have the first transcribed nucleotide, the second transcribed nucleotide, but the 5' cap essentially consists of this 7-methyl guanosine, and this protects the messenger RNA from being broken down and actually helps then the ribosome to attach to begin translation. So remember we had that special group which is kind of found at the 5' end. And the poly A tail, this is at the 3' end. So here we find it at the 3' end of the messenger RNA molecule. So if the sequence contains the polyadenylation sequence, then the RNA is cleaved, and the polyadenylate polymerase adds then 100 to 200 adenine nucleotides to the 3' end. So this creates then the poly A tail, which is really important to transport out of the nucleus, and also adds towards the stability and also translation. Kind of now moving on to gene expression in prokaryotes. So transcription and translation, as we realize, requires a lot of energy, and this can come from the form of like ATP or GTP. Prokaryotes conserve energy and resources by making certain proteins only when they're needed. And in fact, prokaryotes shut off the supply of unneeded proteins. So they help to downregulate the transcription of messenger RNA for that protein. They help to hydrolyze the messenger RNA after it's made, thereby preventing translation. They also prevent translation of the messenger RNA at the ribosome, and they also hydrolyze the protein after it's made, or even inhibit the function of the protein. And prokaryotes tend to use the control of transcription for regulation. And in fact, they downregulate the transcription of messenger RNA for that particular protein, moreover than the other mechanisms to control transcription. So gene expression in prokaryotes, there's a kind of decision at which genes activate involves kind of two types of regulation. So we first, on the left here, we've got negative regulation. So here, negative regulation essentially is the binding of a repressor protein. Okay, so we have at the 5' sequence a site where the repressor protein can prevent transcription. So there's like a binding site. So if we have, if the repressor protein binds, it blocks then the transcription. Okay. Then we also have positive regulation. So this is where you get activation of, so essentially an activator protein binds to the DNA. So normally at a kind of activator binding site. So when you don't have any proteins bound, there is no transcription, and then when you do get the binding of the activator protein, this stimulates the transcription. So regulating gene transcription can actually conserve energy. Well, in E. coli, it's a normal inhabitant of the human intestine. It needs to adjust to the changing environment. And so this means it has to adjust to certain foods. So, for example, you've got certain glucose or certain foods, and those are the availability of that food can change at each meal. And also this presents a metabolic challenge to the bacteria. So, for example, glucose is normally the preferred energy source. So remember, glucose is a monosaccharide. It's really easy to metabolize. And then lactose, it's a disaccharide. So free proteins that are involved in the initial uptake and metabolism of lactose by E. coli are known as the carrier protein, known as beta-galactose permease. We also have another protein which helps to hydrolyze lactose to glucose and galactose, known as beta-galactosidase. And we also have another enzyme called beta-galactoside that helps to transfer acetyl groups. Its role essentially is not clear. So in E. coli, transcription can be regulated. So when lactose is added to the growth media of E. coli, essentially synthesis of beta-galactosidase begins after an initial phase. So remember, so this is like looking at a graph, so looking at the levels of beta-galactosidase, okay, and this is kind of time. So this is in minutes. Okay, so when lactose is added to the growth media, an enzyme essential for the metabolism of lactose is made after the lag period. So this is the lag period, then the enzyme can be made, and there's an induction, and then when you remove the inducer, this will then kind of fall off. So the lag phase is due to the beta-galactosidase messenger RNA synthesis prior to translation. And during this time, so during the lag period, the messenger RNA for the enzyme is made, okay, and then the amount of messenger RNA decreases shortly after the inducer is removed. So the amount of RNA decreases after the lactose is removed, And these changes in messenger RNA indicate that the mechanism of induction is by lactose, which is the transcriptional regulation. So when we think about transcriptional regulation, so we have something called inducers. So lactose actually stimulates the synthesis of a protein, which is known as an inducer. And proteins which are then produced are known as inducible proteins. There are other types of protein which are made kind of at constant rate. These are known as constitutive proteins. So there are many examples in the body of proteins that are required day in and day out, whilst there are some proteins which are not required all the time. And protein synthesis is kind of really a highly enigmatic process, since the kind of the assembly of the messenger RNA, the charging of the tRNA, and the moving of the ribosomes along the messenger RNA all require that hydrolysis of ATP. So it's really kind of very energy hungry. So there are two ways to regulate a metabolic pathway. So here we have our precursor protein, and here we have enzyme 1, enzyme 2, so it makes product A, product B, etc. And the regulation of the enzyme activity. So the end product kind of feeds back. So essentially, when we start to produce more of the product, there's a feedback mechanism from the end of the product. So this is known as a kind of metabolic pathway. And this can actually help to block enzyme activity. This is known as allosteric regulation. The other type of regulation can be where you kind of get a stop, a transcriptional regulation. So there's a stop of transcription of the genes that code for enzymes in the pathway. So this is where we have the end product blocks the transcription of all 5 genes, and no enzymes are then produced. Okay, so moving on to gene structure. We will revisit this again when we consider gene structure, but I just want to introduce it now. So although there's kind of a single stretch of like DNA sequence, the gene is actually composed of these kind of several different parts. So not all of them are encoded in the protein. So what we can see here, this is kind of a simple gene structure overview. Okay, so we've discussed about the promoter sequence. This is normally the regulatory region, so the promoter controls gene expression. And we can have transcription factors which will bind to the promoter sequence. We also have, this is known as an untranslated region, so a 5' end, as the name suggests, it's not translated into the protein. And then we also have, this is known as the ORF sequence, or the open reading frame. This normally encodes the RNA or the polypeptide chain. And then also we have at the kind of the 3' end, another untranslated region, and then we have the termination or the terminator sequences. This is like a regulatory region. It's a terminator which contains sequences that terminate the transcription. So one thing to note is that there are regulatory elements that control gene expression of genes which are upstream, so they're before the sequence, and there are some which control downstream, so such as the regulatory region towards the terminator end of the coding sequence. In prokaryotic gene structure, so prokaryotic genes can be organized into something called operons. So these are also known as polysystromic genes. This is where there are several genes which are under the control of a single promoter. So you just need one promoter, but that will control the gene expression of several genes. And when they're expressed, you get like a single messenger RNA which is produced, which is then translated into a single gene product by the ribosomes. So here we have the regulatory gene region, and now here we have the promoter, and then the operon consists of the promoter sequence, the operator, and the structural genes. And this means that the promoter can actually control the expression essentially of four genes. So the structural genes of A, B, C, and D, which then can produce 4 proteins. So the lac operon of E. coli, essentially, it's a cluster of genes with a single promoter. It's also called the operon. So essentially, we have one promoter, and then that controls gene expression of several genes. The operon that encodes the 3 lactose metabolizing enzymes in coli is called the lac operon. And transcription of operons can be controlled by an inducible operon regulated by a repressor protein. So we can have the binding of a repressor protein. Repressor operon can be regulated by repressible operon can be regulated by a repressor protein, and an operon can be regulated by an activator protein. So it can be activated or controlled by these different types of operons. So the inducible system, so in bacteria, bacteria can switch on and switch off different systems, and lactose, when lactose is absent, we get synthesis of enzymes for its metabolism, which is inhibited. So the repressed protein encoded by a gene prevents the transcription by binding to the operator, and then the RNA polymerase cannot bind to the operator, and you get transcription is then blocked, and then there are no messenger RNA which is produced, so there's no enzyme which is then produced. When lactose is present, so the synthesis of enzymes in the lactose metabolism pathways by binding to the repressor protein and prevents its binding to the operator. So what we see here is when lactose is present, so allolactose induces transcription by binding to the repressor. So when the inducer binds to the repressor, it cannot bind then to the operator. And then the RNA polymerase then binds to the promoter, so then we can get transcription. So the RNA polymerase then can transcribe the genes for those enzymes. So these are known as the enzymes of lactose metabolizing pathway, such as beta-galactosidase, permease, and transacetylase. So when we compare transcription in prokaryotes versus eukaryotics, there are some similarities, lots of differences. Particularly, we have the location of functionally related proteins, so often they're clustered in aperons in prokaryotics, whilst in eukaryotic it's often distanced from one another with separate promoters. So the difference is that we have separate promoters for each of the genes, whilst in prokaryotics they tend to have one promoter which will then control the expression of several genes. In prokaryotics they have one RNA polymerase, whilst in eukaryotic there are 3 polymerases. So we have polymerase 1 which transcribes the ribosomal RNA, 2 which transcribes the messenger RNA, and 3 which transcribes the tRNA, and also those small little RNAs known as microRNAs. The promoters are never regulatory sequences. There are very few in prokaryotes, and there are many in eukaryotes. And initiation of transcription, so you have the binding of RNA polymerase to the promoter, whilst in eukaryotics binding of many proteins, including the RNA polymerase. Now, when we consider how gene is regulated, there are points of gene regulation in the cell. So gene expression can be regulated before transcription. So this is well before transcription. So we can have regulation there. It can be regulated as we're during transcription. So it can also be regulated after transcription, but before translation. So through like processing control. It can also be regulated at translation. So that's also another way, and also after translation. So there is a kind of coordinated gene expression, so both in prokaryotes and also in eukaryotes. And a single environmental signal, so such as a drought stress, so this can cause the synthesis of a particular transcription factor that acts on many genes. So we can take like a repressor, for example, a drought. It activates transcription of a gene that encodes a transcription factor. So what then we get is the binding of the transcription factor to the stress response element, known as SRE. This stimulates then the transcription of several genes, so genes A, B, and C, which then produces different proteins participating in that stress response. And we can also get different types of coordinated gene expression. So, for example, in starvation conditions, where you have reduced calorie uptake, that can induce gene expression in the human body, and there are different genes which help to, which are actually activated in this case. Kind of focusing now on the spliceosome. So we've talked about splicing already. So the spliceosome recognizes introns from exons, and it looks for a certain arrangement of the bases. And particularly it looks for a sequence known as the GU, which is almost always at the 5' end of the start of an intron. And when we look at the 5' end, here we've got a GU region, so a GU sequence within this intron site. And the AG sequence is always found at the 3' end of the intron. So we can actually find these sequences and actually determine where the intron sequence starts and where it ends. The spliceosome actually has the ability then to remove the intron by forming it into a loop or a lariat. So essentially it kind of hoop around, and then it does this by cutting the DNA and then joins the exons together. There's a couple of videos here to have a look at afterwards, if you like. I'll put these up in the Blackboard. So focusing now on the spliceosome. The spliceosome is made up of several RNA and protein complexes called SNRs. So these are small nuclear ribonuclear proteins. And each SNR is a small nuclear RNA, and there are several. There's ones called U1, U2, U4, U5, and U6. Now, so what we can see here is the primary transcript. So here we have our exon. So this is the part that's going to get translated, and we need to kind of cleave out this intron region to then join up the two exon regions. And what happens first is the 3' hydroxyl of the free GMP GDP is used as a nucleophile, okay? And this attacks the phosphodiester band, so the phosphodiester bond between the U and the A. Okay, so between the U and the A. And this, what it does, it releases this first portion of the U ending intron. And then the 3' hydroxyl group, okay, so the 3' of the hydroxyl group of the U ending exon then attacks the 5' end of the adjacent exon. And so what happens then is this then gets kind of spliced out. And then we get the joining up of the ends of those exons. So you get the joining, rejoining of the phosphodiester backbone. Now we can have a look. So this is quite amazing. So in an electron micrograph in yeast, so this is the ribosomal RNA gene. So here we've got the DNA, which is is looking at what's happening under transcription. So what you can see here, this line here is the DNA. So this is where it begins, okay? And what we can see here, this is the RNA. So the RNA which is binding to the DNA. Okay, so as transcription begins, what you get is these little tails coming off, and you can see how much is actually coming off. So these are all the RNA molecules, which essentially there's like smaller RNA tails at the beginning of the transcribed region. So one way to control gene expression is through something called DNA methylation. So what DNA methylation is? DNA methylation is the addition of a methyl group to the cytosine, and so here we have cytosine, and then we have the addition of a methyl group to the cytosine. And there's a reversible formation of the 5' methyl cytosine in DNA that can alter the rate of transcription. So here we have the DNA methylase, which catalyze the formation of 5-methyl cytosine, normally at something called the CpG regions. So these are regions which are rich in C and Gs, okay? And we get transcription is then repressed. So when you get methylation, this actually stops transcription. And this kind of alters the rate of transcription. So this is one way that the body turns on and turns off different genes. Anyway, finally, there's some further reading to take a look at this week. So we have a chapter in Morris et al. In the third edition. So have a look at chapter 16, which is looking at the regulation of gene expression. There are also a couple of videos to take a look at, and also there are two nice papers that you could look at. So this is a messenger RNA journey to the cytoplasm, attire required, and also alternative splicing of genes as an approach for generating protein complexity.